All right, welcome back. We're into 8.3. Let's start with a little review. So the enzyme that synthesizes, that's a fancy word for makes, a new strand of DNA nucleotides from an existing DNA strand is called A, DNA polymerase, B, reverse transcriptase, C, DNA ligase, or D, RNA polymerase. Go ahead and pause the video, think about that. Okay, so I want you to think about this one. We are making a new strand of DNA nucleotides, so there's a good chance that the letters DNA are going to be in the answer there. And then think about what's happening here. So DNA ligase, that is ligating or sticking DNA molecules together. Close, but not quite. DNA polymerase makes polymers, which are long repeating chains of DNA. So that is the best answer. The answer would be A, DNA polymerase. We haven't talked about reverse transcriptase. We'll touch on it slightly. And we haven't talked about RNA polymerase, but in this chapter we will. Can you guess what kind of molecule RNA polymerase makes copies of? That's right, it makes RNA polymers. So DNA polymerase makes DNA polymers, RNA polymerase makes RNA polymers. We'll see that in the gene expression chapters. So chapter 8.3 is all about gene expression. So we take the information from the DNA, we're going to convert it into RNA, which is an intermediate, and then that's going to get translated into a protein. We will talk briefly about the different kinds and functions of RNA. Not too much, that's more of a cell biology thing, but there are different types of RNA. We'll mainly talk about mRNA, which is the intermediate from DNA to mRNA to protein. We will talk about transcription and how it relates to translation, particularly in bacteria. There are some differences between eukaryotic transcription and translation and bacterial. Then we'll talk about the ribosome and the steps of translation that the ribosome goes through. And obviously, again, these critical things are targets of antibacterial agents. All right, so this is our central dogma of molecular biology, right? We have DNA that gets transcribed into RNA. Remember, in the DNA, there are regions that we call genes. The genes contain the coding information that is going to code for a protein at the end. So the DNA gets transcribed into mRNA. That mRNA then gets translated by a ribosome into an amino acid chain. Finally, that amino acid chain folds into the fully developed protein. All right, so here's kind of the overview of gene expression, the process of turning a gene on, which ends up making a protein at the end. The majority, and I say the majority because there are exceptions, of course, is biology. The majority of genes get transcribed into messenger RNA, which carries the information that codes for a protein. That protein is made by the ribosome, so the mRNA message is translated by a ribosome. In bacteria, they have really compact genomes, so they often have multiple genes that are right next to each other, and they actually all get made in one big long mRNA. This is called a polycistronic operon. Poly meaning many, cistrons meaning strands here, or genes, and this operon idea. So in an operon, all the genes are generally related to one another, and they all kind of work together to do something. We will see an example later called the lac operon that is like the gold standard of talking about gene expression and gene regulation. So in here, there are 1, 2, 3, 4, 5, 6 different genes. one of those genes, MDTA, codes for probably a multi-drug transporter A, because we're not very original. There's MDTA, MDTB, MDTC, probably the order they were discovered in. This codes for a gene, for a protein that is a drug efflux pump. So it's a pump that would pump antibiotics out of a bacterial cell. If we zoomed in on this diagram, we could see in here that there is DNA that is what's called codes for the protein. And we'll learn how to decipher this, but in the end of this section, you will know how to read this piece of DNA. That DNA gets turned into mRNA by transcription. So we're going to use one of these DNA strands as a template to make an RNA here. That RNA then goes out and finds a ribosome, which translates it into a string of amino acids, which is our protein here. So let's take a look at an animation about operons. This one is actually weird. It's like hidden in the textbook's animation page, so I'm going to show it here.
An operon is a series of genes in tandem that are controlled by a single promoter. Gene expression begins when RNA polymerase binds to the promoter and transcribes the genes, all into a single mRNA molecule. The genes of an operon typically encode proteins that work together. Here, MDTA, MDTB, MDTC, and MTDD encode proteins of an efflux pump that exports antibiotics such as novobiocin from the cell. Gene BAES encodes an environmental sensor, and BAER encodes a regulator that binds to the promoter and upregulates the expression of the genes in the operon. The DNA sequence specifies messenger RNA and proteins. A variety of sequences in the DNA and mRNA define the landscape of an operon. Here is a closer look at an operon with two genes, A and B. Both strands of the DNA are shown. One DNA strand is the sense or coding strand. It contains a base sequence identical to the mRNA produced from the operon, except that DNA contains the base thymine in place of uracil. The opposite strand is the complementary template strand. RNA polymerase binds to conserved regions of the promoter called the -10 and -35 regions. Binding here directs the polymerase to start transcription just downstream at nucleotide +1. The -35 and -10 sequences are 35 base pairs upstream and 10 base pairs upstream of the transcription start site. RNA polymerase creates an mRNA strand complementary to the template strand. At the end of the operon, a transcription terminator region causes the RNA polymerase to detach from the DNA. Portions of the mRNA are translated into proteins. Each coding sequence of the operon begins with a start codon and ends with a stop codon. At either end of the mRNA are untranslated regions, the untranslated leader and the untranslated trailer. Ribosome binding sites are also untranslated. They direct the ribosomes to the correct locations for translation to begin.
All right, so that animation went into a little more detail than we needed, but it has all the pieces that we're going to talk about. And we will do some exercises in the lab as well that will help us better understand this using some hands-on models. So in the DNA, as it mentioned, there are regions that we call genes. These have the information that codes for a protein. The first step of expressing this information, turning it into a protein, is what I mean by expression, is transcription. So transcription is the process of taking the DNA, using the DNA as a template, as something to build off of, to make an mRNA. The enzyme that transcribes DNA into RNA is called RNA polymerase. And RNA is very similar to DNA, if you remember, but instead of T, RNA has uracil in it, this U. Don't ask me why. It's evolution. It doesn't have to be clean or make a lot of sense. It's just the way the system works. And RNA is single-stranded. It has the sugar ribose in it instead of deoxyribose. So that single-stranded nature is a big difference here, and that means RNAs can fold and do some other things that are really interesting, but it makes RNA less stable. That is why we believe that DNA evolved to become the genetic material that stores everything. There are some hypotheses that RNA was the original kind of genetic material because it can do some different reactions and stuff like that, but DNA, being double-stranded, is much more stable than RNA. RNA is very unstable, so it's kind of an intermediate here. The DNA template, as it's called, you can see can be used to make this mRNA. So if we go A, U, G, A, you can see if we use this bottom strand as a template, we can build a new RNA off of it. And just like DNA, RNA gets built in a 5' to 3' direction by RNA polymerase. So the coding strand up here, the one we call the coding strand, doesn't really do anything. It just sits there. It will read like the RNA that we're going to make, but it doesn't actually do anything. The template strand is the one that's used to build. So you remember, bases pair with one another, A with T. In the case of RNA, U pairs with A. So A, T pairs with T, U pairs with A, G pairs with C, A with T, A with T, A with T, so on and so forth. We can use this information to build this mRNA messenger. How this works in the process of transcription is there's a big complex called RNA polymerase, and if you want to be precise, there's an RNA polymerase holoenzyme, which is all the accessory things that are involved with it. That goes and scans along the DNA. So RNA polymerase is first going to join up with this thing called a sigma factor. It's a small protein that helps find and bind to correct promoter sequences. Remember, promoters are little bits of DNA in front of genes. And the sigma factor it's looking for a specific sequence. So the RNA polymerase kind of hops along the DNA, scanning until it finds two sites. We call it the -35 and the -10. If the sigma factor recognizes those, it binds tightly to the DNA. It grabs onto the DNA. So transcription is actually going to start a little bit further down. But first, we got to find the right place to latch onto. RNA polymerase latches onto here. Now, these sigma factors, there are different ones, and this is somewhat unique to bacteria, the sigma factor itself. They recognize different promoters, and there's different sigma factors for different life stages. So when bacteria are going to divide, there's a sigma factor that goes out and finds all the genes that are necessary for bacterial division. When bacteria need to regulate their flagella, there's a sigma factor for that. When they experience heat stress, there's a sigma factor that goes out and finds genes that will respond to heat stress. When they need to assimilate nitrogen, right? So on and so forth. So there are different sigma factors that will find different genes for different conditions. This is a form of gene regulation. Maybe the immune system is trying to attack the bacteria. There's a sigma factor that will find genes that promote immune system evasion. So there's different sigma factors for different areas, and so there's different sigma factors for different conditions, and we call this gene regulation, right? Turning genes on and off depending on when they're needed and when they're not needed. We'll come back to that in a later section. But I just want to show you what this might look like. So we have RNA polymerase and the different sigma factors that can associate with it, right? They're color-coded them here. They're going to go out and recognize different sequences. So the green one will find green promoter sequences in the DNA. The purple one will find purple promoter sequences, right? The red one, the red sequences, the yellow. So they can go and find different genes and tell RNA polymerase, hey, I need this gene to be made right now. Once that's bound to the -35 and the -10, the sigma factor actually falls off and transcription elongation starts. So the RNA polymerase starts to open up the DNA and copy the template sequence. It starts building a new RNA from a 5' to 3' direction. This is going to incorporate what are called RNTPs, right? We talked about in DNA replication, DNTPs. RNTPs are the A, U, G, and C that get added. And it matches the gene sequence based on base pairing, right? A with U, G with C, that kind of thing. Of course, right, this can be described in a series of steps. For transcription, you have initiation, elongation, and termination. Now, this is where it gets confusing. People confuse this with the DNA replication. This is just a fancy way of saying it starts, it continues, and it ends. The starting of transcription, that's when the sigma factor joins RNA polymerase and they scan along the DNA to find a promoter they bind to. Then once it's found the promoter, the sigma falls off and it opens up the DNA and starts making an RNA molecule off of the template DNA. Finally, when the RNA polymerase reaches what we call a pause site, which is just the end of the gene, some things happen, usually like the RNA folds back on itself or something like that, and RNA polymerase falls off of the DNA and transcription is ended. So we start, we continue, we end. That's the basics. And then there are different things that happen at each of these. Let's go ahead and watch an animation of this.
The first step in gene expression is to make an RNA copy of a gene's DNA sequence. That is, to transcribe a gene. Transcription includes the stages of initiation, elongation, and termination, and is performed by an enzyme called RNA polymerase. In bacteria, before initiation, RNA polymerase includes a number of subunits, including one called sigma. Sigma factor binds tightly to promoters, which target the polymerase to a gene. With the sigma factor attached, the composite RNA polymerase is known as a holoenzyme, meaning that it includes all the subunits needed for initiation. The holoenzyme form of RNA polymerase binds loosely to any sequence of DNA. Thus, within the cell, the RNA polymerase binds and comes off DNA rapidly, as if scanning the sequence. When the RNA polymerase binds to a promoter sequence, however, the binding is tight. To start transcribing a gene, the RNA polymerase opens the DNA, creating a transcription bubble. Now the RNA polymerase can fit a ribonucleotide to base pair with the DNA. So begins the elongation stage of transcription. To elongate the chain, the polymerase catalyzes the addition of new ribonucleotide triphosphates, RNTPs. Each incoming RNTP condenses with the 3' hydroxyl of the preceding nucleotide, releasing pyrophosphate in a step that yields energy. Only a few ribonucleotides can be added before the polymerase stops, because the whole complex is still tightly bound to the promoter. However, now the sigma factor falls off the complex, allowing the core RNA polymerase to continue to move along the template, synthesizing RNA at approximately 45 bases per second. Finally, transcription must finish at the end of the gene, at a pause site in the DNA sequence that causes the RNA polymerase to fall off. In bacteria, even while transcription is occurring, the next stage in gene expression, the translation of a protein, also begins. Here, RNA polymerase is transcribing a gene. The beginning of the growing mRNA has a region called a ribosome binding site, where a ribosome can bind and start translation. As the mRNA lengthens, additional ribosomes can hop on, creating a polysome, which is an mRNA with a series of ribosomes translating the sequence into proteins. This electron micrograph shows a polysome. Notice how the ribosomes farthest from the 5' end, the beginning of the mRNA, show the longest growing peptide chains.
All right, so I think that animation illustrates what's going on quite nicely. Now, it's our turn to practice this. Okay, now we're going to get some practice transcribing DNA into RNA. So I have a double-stranded sequence of DNA here, and you are going to transcribe it into RNA for me. I've even labeled which is coding and which is template for you. So go ahead and pause the video. Go ahead and grab a piece of paper and transcribe this RNA for me. Okay, so where to start? So remember, the template strand is going to be the template which we build the mRNA off of. RNA polymerase is going to transcribe using this as the template. So it's going to go 5' to 3' because it's anti-parallel, just like our DNA. The first thing it would do would be to add an A, because T pairs with A, then a C, A, and here's the tricky one, U, because mRNA has U, A, U, G, so on and so forth down the line. So that's all there is to transcribing DNA into RNA. It's tricky to know where to start and when to end. There are complicated rules that are beyond us. So if I ever ask you to transcribe something, it will just be the whole thing here. Once we have our mRNA, it's time to translate it from mRNA into protein. And this is done by the ribosome. The ribosome is a group of proteins and also some RNA called rRNA, ribosomal RNA, that joins together to take the mRNA and translate the message into a sequence of amino acids. In bacteria, they don't have a nucleus, so this process can happen at the same time as transcription. And you can see here, the ribosomes are making little polypeptide chains, these little wavy things coming off of them. And these ones are much longer because they've had more time to translate the message than these ones over here. Okay, so what actually happens, right? Well, RNA polymerase makes RNA, and then the ribosome comes in and reads that and translates the message into amino acids. How do we decode that message? Well, translation decodes mRNA into amino acids using 3 base pair groups called codons. So every 3 bases, that codes for a specific amino acid. And this is a chart called a codon table that shows these 64 different possible codons. So if the first base is a U, right, you go U, then the second base is a U, and if it's another U on the third base, then it codes for Phe or phenylalanine. It's not critical in any way to memorize this. I don't have it memorized. I don't know it. I would give you a codon table if I wanted you to translate something. But there's a couple of rules we need to learn here. First off, there are 4 really specific codons here. The first one is AUG. This codes for the amino acid methionine. But this is also where translation starts. So the ribosome is going to go along an mRNA until it finds the first AUG, and that's where it's going to start translating. So every protein is going to start with the amino acid methionine. Then it's going to continue on in 3 base codons until it finds one of 3 stop codons. So these stop codons, UGA, UAA, or UAG, they don't actually code for an amino acid, and that ends up causing translation to stop. Now, a side note, if I slip into slang and use the term AA, I am not talking about Alcoholics Anonymous or anodized aluminum. I am talking about amino acids. I'm trying to write it out, but sometimes I use shorthand. So AA means amino acid. And that's what all these things are: proline, serine, histidine, glutamine, all of these things. And these are the 3-letter codes. There are 1-letter codes that sometimes get used. Okay, so how does this actually work? Well, our mRNA has bases in it, right? And there are special RNAs called transfer RNAs or tRNAs that help to transport amino acids to the mRNA to match it up and basically decode the codons. So let's look at this tRNA here. It forms kind of this clover leaf shape. On this end is the amino acid attached to it. And on this end is what we call an anticodon. It's the complementary sequence that matches up with a codon. In this case, this would be methionine, and the codon AUG codes for methionine. So the tRNA actually matches and base pairs with the AUG here. This all is taking place in the ribosome. I will show you that in a moment. For right now, let's just learn how to read the code. So this is awesome. You are now going to be able to read DNA and with it mRNA. Okay, so we are going to translate this mRNA, which you transcribed for me earlier, into a protein. So what's the first step here? I'm going to walk you through it. Okay, remember the rule I just told you? What does every protein start with? Every protein starts with a methionine. So we're going to go along and we're going to scan, just like the ribosome, until we find an AUG. So there's an A, no U. AU, oh, no, AUG. All right, awesome. AUG, that codes for methionine. This is where we start. So our first amino acid is met. Okay, what's next? Now we're going to work in groups of 3. We're going to read the codons. So what's the next codon? A. Okay, so we go to our codon table. A. We need to scroll over to AA, and A, that is lysine or LYS. Okay, cool. And then the next group, GCA. So GCA, alanine, ALA, and then so on and so forth. CUC is leucine. And then we get to the special one, UAA. That's one of our stop codons. So when we get to that, we end translation. That's where it stops. Obviously, this is very short. This is 4 amino acids. That would be like one of the shortest proteins in the world. Most proteins are hundreds, if not thousands, of amino acids. So you can see, right, if we look at the DNA, the coding strand reads much like the mRNA. Remember, the coding strand didn't actually do anything, but when we look at DNA, we can read the coding strand and decode our DNA into what proteins it makes. Remember, the template was actually how we made the mRNA, and then the mRNA, so that's transcription, the mRNA gets translated into the protein. All right, how does translation actually happen? Well, it all starts with the ribosome. So the mRNA is out there, and the ribosome is actually made up of 2 parts. In bacteria, we call them the 50S and the 30S subunit. Now, I just mentioned this because you have different ribosomal subunits. So, what does that mean? That means we can target the difference in the bacterial ribosome, and we attack that with antibiotics. The ribosome, right, the subunits are a mix of what's called rRNA and protein. And the first part, the 30S, kind of matches up until it goes along and kind of checks out the mRNA until it finds the AUG. And now if you want to get real technical, the first methionine, the start amino acid that gets added, actually in bacteria it's special. It's called a formal methionine. It's not important for our purposes, but there is all kinds of weirdness going on. Once the 30S matches up the tRNA, this is a tRNA bringing the amino acid in, matches it with the AUG, it calls out for the 50S subunit, the big one, and they both attach. So now we have a fully loaded ribosome that's ready to go. So we started with methionine. Then if we look at the next bit, there's another slot here called the A or acceptor slot. tRNAs are going to fit in there and see if they can match with the next codon. When one matches, it's going to bind, and a bond is going to form between the first amino acid and the second amino acid. This is a peptide bond. Everything's going to shift over. The first tRNA is going to scoot over to the E or exit site, and it will be ejected. The second one's going to move to the P or peptide site where the bond is actually made. And then now we have a new open acceptor site that will check for the next amino acid by the codon. So this process repeats and repeats until we get to a stop codon. There are no tRNAs that code for anything in stop. Instead, what's called a release factor will bind to the stop and cause everything to fall apart. That releases our new protein and releases the ribosome components to go off and translate another mRNA. This is called termination, and that's where the stop codon causes all of this to release, everything falls apart, and they can go find their next target. I think this is going to make a lot more sense in motion, so let's watch the animation.
Protein synthesis occurs in 3 phases: initiation, elongation, and termination. In bacteria, such as E. coli, initiation requires the association of several components: small proteins called initiation factors, the first tRNA, the mRNA to be translated, and the small subunit of the ribosome, also called the 30S subunit. The key event of initiation is the association of the mRNA and 30S subunit so that the start codon, AUG, is centered on the ribosomal subunit. The 50S subunit docks to complete the initiation complex. Initiation uses energy by hydrolysis of GTP. Note that the ribosome has 3 tRNA binding sites: an acceptor, A site, also called aminoacyl site, a peptide, P site, and an exit, E site. The ribosome is ready for elongating a chain of amino acids. Elongation requires an elongation factor called EFTU, associated with GTP, to bind to aminoacyl tRNAs. This complex enters the A site. Correct selection of the tRNA complex depends on codon-anticodon pairing. Here the anticodon CCC pairs with a GGG codon. GGG codes for the amino acid glycine, which this tRNA carries. Next, the ribosome catalyzes the formation of a peptide bond between the 2 amino acids, and the hydrolysis of GTP drives peptide bond formation. The resulting EFTU, GDP, is expelled. An elongation factor called EFG, associated with GTP, binds to the ribosome. The GTP is hydrolyzed, providing the energy to ratchet the 50S and 30S subunits ahead 1 codon. This maneuver opens up the A site, and the uncharged tRNA slides into the E site. The next aminoacyl tRNA that enters the A site creates a conformational change in the ribosome that telegraphs through to the E site and ejects the uncharged tRNA. These elongation steps repeat along the mRNA. Eventually, the ribosome arrives at the end of the coding region, marked by one of 3 possible stop codons. This phase of translation is called termination. No corresponding tRNAs exist for stop codons. Instead, a protein called a release factor, which has the general shape of a tRNA, enters the A site. The release factor triggers hydrolysis of the peptide-tRNA bond, thus releasing the peptide. The 30S and 50S subunits fall off the mRNA and separate in the cytoplasm, available once again to form a new translation complex with another mRNA.
All right, so hopefully that better illustrates what's going on in translation with the ribosome. Okay, so how does this matter to us? Well, let's talk about our mRNA vaccine, which we mentioned previously, but now I think we have a better understanding of what's going on. So vaccines are a way to pre-train your immune system to recognize pathogens. In this case, let's say this is the SARS-CoV-2 virus, which causes COVID. Our vaccine is going to pre-train our immune system to recognize the virus so that when we are exposed to it, we attack it much quicker and we get much less sick from it. One way that we found to make a vaccine is to expose our immune system to the proteins that are on the virus, in this case, the spike protein. Proteins are tricky to work with. They're hard to get into the cell. What somebody figured out was that if we could get the mRNA that codes for the spike protein into human cells, the human cells will just make that spike protein. On its own, the spike protein doesn't do anything. It's not harmful to us, but it's the main thing that our immune system recognizes. So, we can make RNAs quite easily. We can make them in the lab very, very easily. So, we take the sequence of mRNA that codes for the viral protein, we package it up in a little lipid nanoparticle, and remember your membrane is made of lipids, so this lipid nanoparticle will fuse with your membrane and the mRNA goes inside. Then your human cell, usually a muscle cell in your arm, will take the mRNA and go, I know what to do with this. It will send it to a ribosome, where the protein is translated. Those proteins get stuck on the outside of the cell, and immune cells come along and go, hey, this looks like a virus. I'm going to make defenses against it. And that pre-trains our immune system to recognize the virus, all using this translation side of things. So very clever use of mRNA there. And we can rapidly change these. We're having new mRNA vaccines coming out to different COVID variants. So every time the virus evolves, we can change the mRNA sequence very, very easily and change our vaccines to have the different protein on them. So like I mentioned, obviously, this is a whole ripe target for antibiotics. The 30S and the 50S parts of the ribosome, those are huge targets for antibiotics. So you've probably heard of these tetracyclines are a big one, streptogrammins are another one, aminoglycosides. These are all antibiotics that are targeting the ribosomes there. So if you stop translation, you stop microbes from growing. And you can see some of them are super specific. Aminoglycosides like streptomycin, they target aerobic bacteria, gram-negatives, gram-positives. Those are common enterics or skin pathogens, things like that. So very, very cool use of our understanding of the ribosome and its differences from our own. Okay, so there's a lot in there. This is gene expression, right? Transcription is the first part. That's converting DNA into RNA. That's transcription. So in front of a gene is a sequence called a promoter. The RNA polymerase with its sigma factor scans along until it finds it and binds to the promoter, and then transcription occurs. The RNA polymerase continues transcribing DNA into RNA until it reaches this pause site where RNA polymerase falls off. We then have our mRNA that goes out and finds a ribosome, which translates the RNA into protein. tRNAs, a special type of RNA, decode that mRNA in 3 base codons, bringing amino acids into the ribosome and matching up with the mRNA and adding the amino acid to the growing chain that will become the protein. Translation starts when the ribosome finds the first AUG, the start codon, which codes for a methionine. So all proteins are going to start with methionine. Translation continues, ratcheting along in those 3 base codons, adding amino acids. Remember, there are 3 sites, the A, the P, and the E. So acceptor, peptide, where the bond gets made, and then the exit, where it ejects the spent tRNA. It works in 3 base groups called codons, and there's a codon table to help translate. When it reaches a stop codon, of which there are 3, the ribosome dissociates and falls apart. And the peptide, the amino acid chain that's been made, goes off and needs to fold into its final conformation or shape. All right, that's it for 8.3.